Let me see if I can share my screen. Thank you for inviting me to share with you our program and the progress we have made in our studies. Reneo is a young clinical stage pharmaceutical company. We were founded about 3 years ago, and we're focused on developing therapies for patients with genetic mitochondrial diseases. Our model is to get drugs that big pharma developed for a large indication and that they abandoned development for business reasons. We take those drugs, we adapt them, and use them to treat rare diseases. And this is all based on understanding the mechanism of action, the biology of the drug, and whether it can be used to treat other diseases that were originally intended for. The advantage of doing this is that by the time we acquire the drug, we already know a lot about the drug and how it works. We have a fairly extensive safety and pharmacology package. We have done advanced work in understanding the chemistry, the manufacturing, and the quality of the drug. And in the case of REN001, in addition, clinical trials in healthy volunteers had already occurred. So we have a very good understanding of the PK, that is how a drug is absorbed, distributed, and eliminated from the body. And the drug has been tested in early studies. So we have an advantage of being able to see what is the pharmacodynamic effect of the drug and what is the potential dose that one wants to use moving forward in the indication we want to pursue. So REN001 is a PPAR delta agonist, and PPAR delta agonists are nuclear, are drugs that stimulate a nuclear receptor. The PPAR delta is a nuclear receptor that is primarily present in muscle cells. It can also be found in smaller quantities in other tissues, such as kidney or liver, for example. But the primary manifestation of PPAR delta is in muscle. And this receptor, this nuclear receptor, PPAR delta, controls genes involving cellular energy metabolism. Activation of PPAR delta increases the enzymes in mitochondria that are involved in whole body fatty acid oxidation. And it also activates genes involved in the formation of new mitochondria or mitochondrial biogenesis, and it increases oxidative muscle fibers. All of these effects may be of much help to patients with mitochondrial diseases. A unique aspect of the mitochondrial diseases is the fact that in some patients, the patients that have a mitochondrial gene defect, there is heteroplasmy. And heteroplasmy means that in any given muscle, cells can have normal and abnormal mitochondria, and the number of normal and abnormal mitochondria can determine how severe the disease is going to be. So we aim to increase the absolute number of normal mitochondria, and we also aim to increase the function of the normal or perhaps even the abnormal mitochondria. And we have done experiments in mice and in cellular cultures and in humans. And I'm going to share a little bit of what we have so far. In the mice, after a single dose of REN001, we looked at their muscles to see changes as they occur, measuring messenger RNA, and we saw increases in PGC, for instance, the master regulator of mitochondrial biogenesis. We also saw increased expression of activation of other genes like CPT1, ALAS1, and CYCS, all of which are involved in fatty acid oxidation. In a study in humans that was done in healthy volunteers, we looked at the effect of REN001 to preserve muscle function. And this actually was done by the company that had the drug before we acquired it. They were trying to explore whether REN001 could be used to treat patients who are ventilated for prolonged periods of time in order to preserve muscle mass and muscle strength. And what they did in this particular study is they took volunteers and they had one leg immobilized with a cast for 14 days. And as you know, immobilization results in muscle atrophy and weakness. Half of the patients received REN-001 and the other half were given a placebo. The patients that received REN-001 had more strength in the leg after the cast was removed, and they recuperated faster than the patients that received placebo. I'm not showing you data here, but before and after biopsies were obtained, and what was found is that there was activation of the genes involved in mitochondrial biogenesis in the muscles of the patients that received the drug. So based on these findings, from our animal studies and everything that we've done so far, we've considered many diseases where a PPAR delta agonist can be useful, and I've outlined most of those diseases in this graph. We are a small company, and we are focused on efforts in three main groups of diseases. We've recently completed a proof of concept study in primary mitochondrial myopathy. We initiated a study in patients with long-chain fatty acid defects, and you can see those defects outlined there, CPT2, LCHAT, BILCAT, trifunctional protein defects. And we've also initiated a study in patients with a glycogen storage disease type 5. The McArdle patients or glycogenosis type 5 is a disease where patients cannot use glycogen in the muscle, and they depend on mitochondria to process fats and supply energy. And we believe that enhancing this pathway may benefit these patients. Now, we have completed the proof of concept trial. We have completed the concept trial in PMM and initiated studies in fatty acid oxidation and defects in McArdle. And in all of these studies, the drug has been safe and well tolerated. Unfortunately, in the case of FAOD or fatty acid oxidation defects and in McArdle disease, these studies had begun just right before the pandemic began. So these studies had to be temporarily paused due to the COVID-19 pandemic. The McArdle study is already enrolling again in Spain and will soon enroll in other centers. The FAOD studies are about to begin again in the United States. We hope to collect enough data from those studies to be able to move forward. I'm going to focus now primarily on the PMM program. And let me switch a little bit and share with you about the process that we are following to address primary mitochondrial myopathy. PMM is not an easy problem to tackle, of course, and you all know that. When we started thinking about the disease, we decided that it was very important to talk to patients about their disease and gain a better understanding of the problem from the patient's perspective. So we designed a study to interview patients and obtain a list of relevant bothersome signs and symptoms that patients present with. And we also wanted to inquire what are the impacts that are reported by these patients of these symptoms. As part of this study, we also wanted to determine if available questionnaires such as the Modified Fatigue Impact Scale can be used in PMM. So we conducted semi-structured interviews with open-ended questions and that encouraged spontaneous responses by the patients. And we conducted a total of 20 individual interviews with patients with PMM. The mean age of these patients was 37 years old, and there were twice as many women as men, although the diseases manifest with equal frequency in the sexes. Most of the patients that we interviewed were white. Patients reported a wide range of general health status from very poor to very good. So that gives us a wide array of patients with different manifestations that enabled us to see what are the main problems that these patients present with. So the most common symptoms reported by these patients were exhaustion, fatigue, and lack of energy. When asked what is the most bothersome symptom that you have, patients told us that the most bothersome symptom was fatigue, exhaustion, and lack of energy. In addition to these symptoms, patients reported many symptoms related to muscles, such as weakness, cramping, spasms, tremors, but they also reported other symptoms such as blurry vision, ptosis, and dizziness. Most commonly reported impacts were that there were limitations on physical activities or exercise, that patients needed to pace themselves to perform physical activity. There was a lack of independence or reliance on others, and needing to rest more often or for longer periods of time in order to complete a task. We also presented the patients with a modified fatigue impact scale that is a brief questionnaire that addresses fatigue in the last 4 weeks. And the patients first told us that the language in the questionnaire was clear and concise, and they could understand it very well. And they also told us that the modified fatigue impact scale largely captures the overall experience of fatigue that they themselves have. So they felt that this would be an instrument that would be appropriate to include in clinical trials. And we are actually including the questionnaire in the trial. So let me talk a little bit about the study we did in patients with PMM. The primary objective of that study was to evaluate safety and tolerability. We also collected data on exercise tests and symptom questionnaires. There was an open label. This was an open label study, meaning all patients received the drug. And as you know, mitochondrial diseases come in many flavors. So we enroll only patients who had mitochondrial gene defects. And the reason for doing only mitochondrial gene defects and not nuclear gene defects as well is because we really don't know what the mechanism of action of our drug will be in patients. And we cannot exclude the possibility that the main activity may be biogenesis and increase of mitochondria. And if that is the case, we would like to be in a position where we see an expansion of the normal mitochondrial pool. So that's why we have focused on patients who clearly have heteroplasmy. Patients received REN-001 for 12 weeks in Part A and had the option to continue treatment in an extension study. The majority of participants who completed Part A elected to continue treatment in the extension. Now, here's a list of the outcome measures we included in the study. We had a walk test. We did gait analysis, peak exercise tests, and submaximal exercise tests. We don't anticipate that in future studies we will include all of these exercise tests, but we wanted to make sure that the more controlled studies such as gait analysis, peak exercise, and submaximal exercise test would help us validate the value of a walk test that can be implemented in a larger population of clinics. The questionnaires that we did focused primarily on fatigue and pain, but we also included the SF-36, that is a quality of life instrument, and includes subscales of fatigue and energy as well. The study was a small study. We enrolled only 23 patients, 17 completed the study, and 13 of the 17 that completed the study went on to the extension study and completed almost, the patient that was treated the longest was treated for almost 40 weeks. Now, only 17 patients of the 23 completed the study, and this is because of the COVID pandemic. It began in the middle of the study, or almost at the tail end of the study, and some patients were not able to continue in the trial because they were ordered to stay home. This study was done in England, in the UK, and and we had to suspend the trial at that time, and we lost 5 patients that were participating in the trial due to the COVID pandemic. But in general, what I can say is that REN-001 was very safe and well tolerated in the patients with PMM. We saw clinically important improvements in the walk tests and in the other exercise tests. We saw meaningful improvements in symptoms and quality of life questionnaires. And the majority of patients who took part in the study chose to continue treatment in the extension part. The data that we obtained was sufficient for us to inform the design of the next study. And we still have a few parts of the study that are being evaluated. For instance, the muscle biopsies that were done before and after treatment are just now being evaluated because the pathologists were very busy with the COVID pandemic. So final results of the trial are not available, and that's why I'm not more specific about the results in the study. Those will be presented at a later date. Now, a key element in designing a successful study in patients with PMM is finding patients with similar characteristics. It's addressing the heterogeneity of the disease. So patient selection in our mind is very important in achieving success. The next study will again focus on patients with mitochondrial gene defects and exclude patients with nuclear DNA defects. We plan to evaluate patients who have predominant myopathic symptoms with impaired performance in walk tests. And there will be a patient selection oversight committee that will review all patients prior to acceptance into the study. We plan to complete a walk test at screening also to reduce the training and learning effect. And this is very important because it's well known that in all exercise tests, the first test that patients do typically is not as good in performance as the next test. So there is an element of a learning effect that occurs when you do for the first time a walk test, where you feel more comfortable performing the test and then you do better in subsequent tests. So in order to address that problem, we're including in the screening period a walk test to try to familiarize patients with the performance of the test. So moving forward, these are our plans. We're preparing to start a 24-week double-blind placebo-controlled study. The study will enroll about 200 patients, and the patients will have primarily mitochondrial gene defects. This will be a study for adults 8 years and older with a history of myopathy predominantly. The study will include about 30 centers in North America, in Europe, and in Australia. And we plan to include walk tests and symptom questionnaires in the study as endpoints. We have done most of the groundwork to get the study approved in multiple countries. That includes talking to regulatory agencies like BfArM in Germany, the FDA in the US, MHRA in England, etc. And we plan to have a few sites in Italy as well. If all goes well, centers will be ready to enroll patients by the beginning of 2021. It is possible some centers may be able to start earlier, and this all depends on the pandemic, of course. And we're very excited about the progress we've made, and we really look forward to continue to work with the PMM community in order to accomplish the study. I want to thank you for your attention and hope to be able to give you more updates as we progress with our studies in the months to come. And I want to thank the organizers again for giving us the opportunity to speak in this meeting. Thank you.